viewers, Assalamu alaikum. We are studying the life cycle of ameloblast. According to their function, the lifespan of the cells of the inner enamel epithelium can be divided into the 6 stages. Morphogenic, number 2 organizing, number 3 formative, number 4 maturative, number 5 protective, and number 6 the desmolytic stage. Since the differentiation of the ameloblast is most advanced in the region of the incisal edge or tips of the cusp and least advanced in the region of the cervical loop, all or some stages of the developing ameloblast can be observed in the one-tooth germ. Stage number 1, the morphogenic stage. Before the ameloblasts are fully differentiated and produce enamel, they interact with the adjacent mesenchymal cells, determining the shape of the dentino-enamel junction and the crown. During this morphogenic stage, the cells are short and columnar, with a large oval nuclei that almost fill the cell body. The Golgi apparatus and the centrioles are located in the proximal end of the cell, whereas the mitochondria are evenly dispersed throughout the cytoplasm. During ameloblast differentiation, the terminal bars appear concomitantly with the migration of the mitochondria to the basal region of the cell. The terminal bars represent points of close contact between the cells. They were previously believed to consist of the dense intercellular substance, but under the electron microscope, it has been found that they comprise thickening of the opposing cell membrane associated with the condensation of the underlying cytoplasm. The inner enamel epithelium is separated from the connective tissue of the dental papilla by a delicate basal lamella. The adjacent pulpal layer is a cell-free narrow light zone containing fine ergyrophil fibers and the cytoplasmic processes of the superficial cells of the pulp. Stage 2, organizing stage. In the organizing stage of development of the inner enamel epithelium, interacts with the adjacent connective tissue cells which differentiate into the odontoblast. This stage is characterized by a change in the appearance of the cells of the inner enamel epithelium. They become longer and the nucleus-free zones at the distal end of the cell become almost as long as the proximal parts containing the nuclei. In preparation for this development, a reversal of the function polarity of these cells take place by the migration of the centrioles and the Golgi regions from the proximal end of the cell into their distal ends. Spatial staining methods reveal the presence of the fine acidophil granules in the proximal part of the cell. Electron microscope studies have shown that these granules are actually the mitochondria, which have become concentrated in this part of the cell. At the same time, the clear cell-free zone between the inner enamel epithelium and the dental papilla disappears, probably because of the elongation of the epithelial cells towards the papilla. Thus, the epithelial cells come into close contact with the connective tissue cells of the pulp, which differentiate into the odontoblast. During the terminal phase of the organizing stage, the formation of the dentine by the odontoblast begins. The first appearance of the dentine seems to be a critical phase in the life cycle of the inner enamel epithelium, as long as it is in contact with the connective tissue of the dental papilla. It receives the nutrient material from the blood vessels of this tissue. When the dentine form, however, it cuts off the ameloblast from their original source of nourishment, and from then on they are supplied by the capillaries that surround, may even penetrate the outer enamel epithelium. This reversal of the nutritional source is characterized by the proliferation of the capillaries of the dental sac and by reduction and the gradual disappearance of the stellate reticulum. Thus, the distance between the capillaries and the stratum intermedium and the ameloblast layer is shortened. Experiments with the vital stain demonstrate the reversal of the nutritional stream. Stage 3, the formative stage. The ameloblast enter their formative stage after the first layer of the dentine has been formed. The presence of the dentine seems to be necessary for the beginning of the enamel matrix formation, just as it was necessary for the epithelial cells to come into close contact with the connective tissue of the pulp during the differentiation of the odontoblast and the beginning of dentine formation. This mutual interaction between one group of cells and another is one of the fundamental laws of organogenesis and histodifferentiation. During formation of the enamel matrix, the ameloblast retain approximately the same length and arrangement. Changes in the organization and the number of the cytoplasmic organelles and nucleons are related to the initiation of the secretion of the enamel matrix. The earliest apparent change in the development of the blunt cell processes on the ameloblast surface, which penetrate the basal lamella and enter the predentine. 4. Maturative stage. Enamel maturation, full mineralization occur after most of the thickness of the enamel matrix has been formed in the occlusal or incisal area. Cervical parts of the crown. Enamel matrix formation is still progressive at this time. During enamel maturation, the ameloblasts are slightly reduced in length and are closely attached to the enamel matrix. The cells of the stratum intermedium lose their cuboidal shape and regular arrangement and assume a spindle shape. It is certain that the ameloblasts also play a part in the maturation of the enamel. During the maturation, ameloblasts display microvilli at their distal extremities, and cytoplasmic vacuoles containing material resembling the enamel matrix are present, and these structures indicate an absorptive function of these cells. Stage number 5, the protective stage. When the enamel has completely developed and has finally calcified, the ameloblasts cease to be arranged in a well-defined layer and can no longer be differentiated from the cells of the stratum intermedium and the outer enamel epithelium. These cells layer then form a stratified epithelium covering of the enamel, the so-called reduced enamel epithelium. The function of the reduced enamel epithelium is that of protecting the mature enamel by separating it from the connective tissue until the tooth erupts. If connective tissue comes in contact with the enamel, anomalies may develop. Under such conditions, the enamel may be either absorbed or covered by a layer of cementum. In this phase of the life cycle of ameloblast, the epithelial enamel organ may retract from the cervical edge of the enamel. Adjacent mesenchymal cells may then deposit afibrillar cementum on the enamel surface. The last stage, the desmolytic stage. The reduced enamel epithelium proliferates and seems to induce atrophy of the connective tissue, separating it from the oral epithelium, so that fusion of the two epithelia can occur. It is probable that the epithelial cell elaborate enzymes that are able to destroy connective tissue fibers by the desmolysis. Premature degeneration of the reduced enamel epithelium may prevent the eruption of the tooth. Thank you so much for watching the video. Don't forget to subscribe the channel.